evening. My name is Allen Lin, and it is a pleasure and honor to be speaking with all of you here tonight. First and foremost, I want to congratulate all of the finalists for making it to this stage. Before this camp, you have already excelled in your study of biology through hard work and rigorous study, and especially through the famous Campbell textbook. And now at the finalist camp, you practice various laboratory dissections and practices. I've heard using a Bunsen burner for the first time, dissecting fish, learning chromatography, various microbiology practices. So the dedication you've shown to the field of biology is truly remarkable and to be commended. As Mary mentioned, I was a USABO finalist myself and had the opportunity to attend this camp. Back then it was in George Mason, twice, in 2005 and 2006. I was sitting where you are now, filled with excitement and curiosity. Attending this camp was certainly a transformational experience for myself, solidified my passion for biology and understanding the working of life. And also I made a lot of friends that I kept in contact with undergrad, graduate to now through this camp. So the people that you've met now, you will be seeing them again. After the second time here, I was fortunate enough to go on and compete and win a gold medal in the IBO in Argentina. I also want to express my gratitude of all the counselors, the lecturers, the staff, Mary and Joanne, and the sponsor who made this camp possible. Having been a counselor myself multiple times, I appreciate all the effort that goes into making this camp happen, from leading lessons in front of the students, to also all the work that happens behind the scenes, such as writing and grading exams and coordinating logistics. You must be proud of how you've seen the students grown so much in just 2 short weeks. So I've been asked to speak a little bit about my journeys since being at USABO and to share a few lessons that I've learned along the way. In this camp, you're exposed to numerous areas of biology, from single-cell bacteria to think about entire ecosystems, from observational analyses such as writing flower formulas to learning about advanced sequencing techniques. I encourage you to continue on this quest for knowledge as you progress in your careers. So after my time at USABO, I attended MIT for my undergraduate studies, where I was drawn to the then new field called synthetic biology. This field uses advances in genetic engineering to program cells like logic gates on a circuit chip, and to use these cells to potentially address medical and environmental challenges. My first research experience was participating in this interesting team-based competition called iGEM, which you will hear about in your own colleges once you get there, where undergrads work collaboratively on summer projects in this field of synthetic biology. I remember my first research experience was being part of a team at Caltech where we engineered human gut bacteria to defend against pathogens and treat lactose deficiency. It was this experience of being able to engineer live organisms to do things that they hadn't done before that really got me hooked onto the promise of synthetic biology and this idea that we're now entering, even back then, a stage where we can quote-unquote program life. So as an undergrad, I spent my time during the school year studying chemical, biological engineering, with computer science, and during the summers I would conduct more synthetic biology research. So that was what I was doing sort of on the science direction as an undergrad. I also want to talk about other things I became interested in as an undergrad, which was while working in this field of synthetic biology, think about how to program life or do new things, I was concerned about the sort of the implications on both the safety and also security side. What does it mean to be able to program cells to do things that they don't naturally do? So this actually led me to my university's political science department, where I worked with this very wise professor called Ken Oy, and also someone who thinks ahead quite a lot about proactive risk management policies for synthetic biology. We thought about the benefits and the risks of technologies, like bacteria-based biosensors for arsenic contamination. And we even had a meeting here at DC to gather experts across various agencies who think about these issues collectively. And so I want to encourage all of you, as you work on sort of the latest technologies, also think about the broader implications, both from policy-wise, societal-wise, regulatory-wise, because I think all these things in that come together and really shape the adoption of these new technologies. I'll speak a little bit about after my time in synthetic biology. After that period of my life, I went on to the UK, where I received a Marshall Scholarship to study technology policy and public health. And speaking briefly about my time in public health, I just want to say that I spent some time focused on this interesting field called health economics, where the question there was, how do you maximize limited healthcare resources to make the largest impact in terms of producing the most number of quality life years that people can enjoy. There, I had the pleasure of working on looking at the effectiveness and potential expansion of HPV vaccination to gay and bisexual men. Back then, the UK was only vaccinating females. Quite exciting that we're now in a period of time when we have so many tools to not just read, but also to edit, to delete, and even write DNA or RNA into cells. We have technologies like CRISPR-Cas9 and all of its variants. We have lipid nanoparticles, so all the COVID vaccines you got use LNPs. We have RNA interference in the form of sRNAs. So with this explosion of different genetic modalities, I think it's a fantastic time to be entering the space of biology, and also particularly excited about the fact that all of these new genetic medicines makes drug design more amenable to machine learning methods, because the sequence is the drug itself. So think about, there are over 5,000 non-genetic diseases that affect about 1 in 100 people. And with these new DNA and RNA technologies, we might be able to not only understand these diseases, but also fundamentally cure the root cause of these non-genetic diseases. I cannot imagine a better time to be working in biology, and I think part of the joy of being in the life sciences is that the work we do could eventually help improve the lives of people, and this has always served as a very powerful motivator for me. So I hope these experiences have given you some insights into the types of directions one can go after this camp, and we're generally in the field of biology and life sciences. And I want to close with 3 brief thoughts. First, try and get yourselves into the intersections of different disciplines. Many breakthroughs occur when a new technology is applied to a new field. Second, keep an open mind as you progress in your academic journey. Right now, you want to explore widely, and you might surprise yourself. For example, me being very STEM-focused in high school, I would not have guessed that I enjoyed thinking about policy and regulatory frameworks. Lastly, remember that doing new research is a team sport. If you look at many of the major academic papers, you'll see a very long list of co-authors, and that's what it takes to do science, especially one that is very complex in this age. So it's important to also develop the skills to work effectively with others. People can be complex, and learning to collaborate well can have a significant advantage and influence on your effectiveness and also on your personal fulfillment, surprisingly than the specific type of science you're doing. So with these thoughts, getting into intersections, staying curious, and understanding that doing science is a team sport, I want to congratulate all the finalists once again on your remarkable achievements. Your hard work, dedication, and passion for biology have brought you to this significant milestone. My bias is that now is the best time to get into biology, given the advances in genetic engineering and genomics. But regardless of the fields that you pursue, the skills you developed here will serve you well